Good. And then from there, we're going to go to the epithalamus. So the epithalamus, here's the thalamus, hypothalamus, epithalamus is in the back. There we have mostly the pineal gland, which we discussed in the endocrine system, and the choroid plexus. And the choroid plexus, we'll talk about that here. That's that red stuff right in here that you can see. The red stuff that are actually lines, these ventricles, and ventricles are deep chambers that reach into the brain tissue itself and are filled with fluid. They're actually filled with cerebrospinal fluid. And the cerebrospinal fluid is made or secreted from the arteries that reach into those chambers, and that's that red stuff. And when you look at those little capillaries, you see one coming in right here. You have cells around it, and those are actually neuroglial cells. They're support cells of the nervous system, and they make sure only certain things that we want goes from the blood to the CSF. Oh, I shouldn't be trying to draw with this stuff, huh? There we go. I should do this. CSF, cerebrospinal fluid. And cerebrospinal fluid is a chemical broth that nourishes the brain as well as gives it buoyancy and effectively reduces its weight by 97%. Buoyancy is really cool because buoyancy counters gravity. So you have gravity on top pushing down. That's the heavy stuff. And buoyancy from here comes up and pushes upward, and so that's when you're floating in the water. That's what that basically means when you explain what's going on when you're weightless. But the brain is so soft that without any watery film around it, it collapses. See here, it's firmly, nicely attached to the skull, so to speak, with of course these membranes around it. But then look at the space it gets right here. And all that other stuff is filled with air if there is no water film around the brain protecting it. Sometimes that happens when we have what's known as a lumbar puncture, where we put a needle in the cord, well, in between the dura mater and the cord, not into the cord. And because that's where in the arachnoid space, subarachnoid space is where that CSF is. And so sometimes we want to take some of that stuff out to have diagnosis of what's going on. Maybe there is, sometimes we have bacteria in there, which is not good. And sometimes we want to bring medicine into that area. For example, when you give baby birth as a spinal tap, and epidural, we can put medicine in there in order for us to not feel all those contractions so badly. The problem is, if we make a hole in the dura mater and the hole doesn't close, we then can have a situation where we leak out the fluid, the cerebrospinal fluid, and don't have enough, and the brain collapses when we're standing up, and that can give people intense headaches. So that's where that becomes important as well, that buoyancy thing. So then internally we have these chambers, and we describe these chambers as ventricles. So we have a few of them. We have two lateral ones on the side, so they're like horns actually reaching. It's not done justice with this picture. They reach lateral into the brain from both sides, like horns, really. And then we got a third ventricle, and that's medial to the thalami, right in between there. And from there, we're going to have a cerebral aqueduct. That's kind of like just a channel, really. But they have to give it a really cool name. Cerebral aqueduct. Cool name. And then we get to the fourth ventricle, and that's in front of the cerebellum that we're going to talk about right here, and behind the pons. And then it goes from there, the cerebrospinal fluid goes to a central canal into the spinal cord, or in between, actually through the spinal cord, deep inside. You can see it down here nicely because you see the pathway of the fluid. So that fluid is made in these choroid plexi, circulates around in the ventricles, and then has a few holes in the back here, these apertures, they call them, and then from there goes into the subarachnoid space surrounding the brain and the spinal cord. And then from there is these granulations, arachnoid granulations. Those are actually vessels that reach, or actually cerebrospinal fluid pockets that reach into the venous blood supply and bring through what's known here as the superior sagittal sinus, which is just another word for something else. That's a venous structure right here, and that drains the CSF and brings it back to the circulatory system. And in the spinal cord, basically the fluid just leaks out on the sides of the inner vertebral foramen. Holy moly, that's a lot of stuff, isn't it? We're not done. Then we get to the midbrain, and that lies between the diencephalon and the pons. So that's a weird structure. That's kind of like right going through here, right around here. That's that structure. But what's interesting there, we have 4 bumps that go on the back end that you can see, if I actually erase what I just did, that you see there in the back end right there and then right there. And these 4 bumps collectively are called the corpora quadrigemini, but they're made out of, and what they do is they coordinate eye and hand movements when we're triggered. So we have 2 of those. We have the inferior colliculi, and they harbor reflex centers that react to auditory stimuli. So we hear something in the periphery, and we have to look at it. So that's that one. And then we have one on top, the supracolliculi, and that does the same thing, but to visual stimuli. So we see something in the periphery, we have to look at it. Is it a bug? Is it an airplane? I don't know, something that's dangerous. And we just then look at it. So it coordinates all that stuff, the eye and hand movements with it. So those are kind of interesting. I remember that the superior colliculus is on top to the inferior colliculus, and my eyes are higher than my ears. That's how I remember which one's doing what. And then we got other cell bodies deep in there. We got the substantia nigra, which is linked to the basal ganglia. That's right here. Oops. Substantia nigra is actually in blue, these blue dots. And then we have the red nuclei, which influence, and that's red, nice, which influence limb flexion motor pathways. And they are collectively part of what we call the reticular formation. And those are nuclei that reach all the way from the medulla oblongata, which is the lowest part that we're going to talk about soon next, and they go all the way up to the cortex, basically, or the diencephalon, right here. And what they do, they want to actually help the cortex be less or more alert. So what they do is they filter out excitabilities of sensory information and make sure not everything reaches the cortex, or filter what reaches the cortex. And that's why we actually have an ability to sleep. If we need to sleep, we don't get all that information coming in. Whew. The cerebellum in the posterior part of the cranium functions mainly to maintain equilibrium and muscle tone. It coordinates the activity of the voluntary muscles. So this is good because that helps us not fall down. So if we, well, that arrow should have gone out, huh? So that helps us not fall down, basically. And then it also coordinates activity of voluntary muscles with the programmed movement patterns in the basal ganglia. So this is what I'm thinking about when I I want to go and turn on the light. And so I have to walk across the room. So that's what I want to do. That's primary motor cortex. And then from there, I start walking. That's a rhythmic motion. That's basal ganglia. And then it's of course dark, and there's something in the room that I trip over, and I almost fall. And the fact that I don't fall is because of the cerebellum. That's why I don't fall. And so that's how I think of that. And from there, we then go into the pons. And the pons means bridge. Oh, that should have been bold, huh? The pons means bridge, and that's anterior to the 4th ventricle. And that basically carries tracks. It carries some tracks that go up and down. So I put them in green here. They call them cerebral peduncles. So cerebral peduncles goes to the top of the head, the cerebrum. And then we have tracks that go in and out of the brain, the cauliflower brain, and those are cerebellar peduncles, not cerebral, but cerebellar. So we got a few different ones. I just put arrows there to indicate where those lie. And lastly, that brings us to the medulla oblongata, and that's then the structure that is a transition between the spinal cord and the brain. A few things we have there. In the front, when we look in the front, we got these tracks here, these things that these things that bulge up a little bit. So these are in the front. And we known those as pyramids because those carry voluntary motor messages from the cerebrum to the spinal cord. So those are pathways going down, not up and down, just down to the skeletal muscles. They're known as pyramids not because the stuff looks like a pyramid, but because the cells the cell bodies where the initial impulse comes from, up in the head, that looks like a pyramid. So that's why. And then the next structure that's interesting is right down here, and that's a place where these fibers cross. The one side goes to the one, and the other to the other side. That's why we have the right side of the brain controls the left side of the body. So that's known as decussation or crossing. Interesting, huh? The olives are gray bulging nuclei that come out laterally, and I put them right here. So this is actually a structure from behind. This is posterior here, posterior. So the olives are right here on the side, and they are involved in passing information about muscle joint stretch to the cerebellum. So what's really interesting always to me is how you can see how much stuff in that brain has to do with moving. So that's why, we don't move, we lose a brain. And when you look at a kid and it starts walking, its brain starts developing like crazy. But deep in here, we also have other nuclei, and those are mostly these visceral centers, like the cardiovascular center that influences how the heart beats or how fast it beats or how strong it beats. And then we have respiratory centers. Respiratory centers are kind of interesting. Some of them need to turn on the breathing in, and then we have a stretch. When there's enough stretch, the system says stop breathing in, and then we breathe back out. So it's a rhythmic thing. So those things also happen down here. All right. Well, that was a lot of stuff. I hope you enjoyed it.